In this presentation, we'll cover introduction to enzymes. So, enzymes are biological catalysts, and they facilitate chemical reactions or mediate transformation of one form of energy into another. Some of the common features of the enzymes are that they have high catalytic power, high specificity, and that means specificity that could be either specificity towards a particular substrate or towards a particular reaction or both. And also, almost all of the enzymes are proteins. There are some relatively few enzymes that are RNAs, ribonucleic acids. So, enzymes are specific, as I mentioned already, for particular substrate and particular reaction. So here are some examples of proteolytic enzymes. Proteolytic enzymes are those that hydrolyze or cleave peptides or proteins. So papain would be non-specific. It basically cleaves any peptide bond. Trypsin is specific, and it cleaves only a chain on the C side of lysine or arginine residue. And thrombin is highly specific, and it will cleave peptide chain only at arginine glycine bond, and only when that bond is present in certain sequences. When we look at structure of an enzyme is typically composed of a protein part, which is called apoenzyme, and a cofactor, which should be not protein component, and it's called coenzyme. And so together they form holoenzyme. Typically we call holoenzyme just enzyme. Now a cofactor, non-protein part, could be either a metal or some small organic molecule, typically a vitamin. If that small organic molecule is tightly bound to the cofactor, then we call that prosthetic group. If it's loosely bound, then it's called a substrate. Or co-substrate. A substrate is what enzyme acts upon. This would be a co-substrate. There are different types of transformations, biological transformations that enzymes are involved in, and one important type are energy transformation. So, for example, enzymes are involved in photosynthesis, where energy of electromagnetic radiation is converted into energy of chemical bonds, where glucose is synthesized, so a molecule that has relatively high energy chemical bonds. And then in the next stage in mitochondria, that chemical bond energy of glucose is converted into ion gradient, and that's also catalyzed by enzymes. And then again, in an enzyme catalyzed process, that ion gradient is used to synthesize ATP. So another molecule that also has chemical bonds of high energy content, and that energy of ATP could be used for various biological processes in the cell, or it could be used to generate mechanical energy, such as contracting muscles. Now, enzymes only affect the reaction rate, not the position of the equilibrium. And that's common to all of the reactions that are catalyzed, biological or non-biological. So without an enzyme or without a catalyst, reaction proceeds at a certain rate. In this case, we have relatively simple reaction where reaction concentration increases linearly with time. Concentration of the reaction product increases linearly with time. Without enzyme, reaction is relatively slow, and with enzyme, we have rather fast reaction, and so the amount of product shown now in red obviously increases much faster, and very quickly we reach a stationary state. So, as I already mentioned, enzymes only affect reaction rate, how fast equilibrium is reached, not the position of that equilibrium. When we consider mechanism of action of enzymes, enzymes act by changing the nature of the transition state. So in a hypothetical reaction where substrate S is converted into product P, there is transition state X, indicated here as X in square brackets with double dagger, as a transition state. And you can see that activation energy necessary for substrate to reach the transition state is rather high. That means that reaction is slow. Enzyme catalyzes the reaction by forming enzyme-substrate complex. So enzyme forms an additional intermediate, adds one more step to the reaction. And so now we have a transition state that leads to formation of the enzyme-substrate complex or enzyme-substrate intermediate. And that transition state is considerably lower in energy compared to transition state needed to reach intermediate or transition state X, not intermediate, but transition state X. That means that reaction is much faster. And then in the second step, enzyme-substrate complex is converted into product. And also that step has its own activation energy that is pretty low. In fact, that one is very low. That's obviously a very fast process. And so in this case, we can see that enzymes act effectively by lowering the activation energy, which means that overall reaction proceeds at much higher rate. The process of formation of the enzyme-substrate complex involves first binding of substrate to the active site of the enzyme. And this active site also contains catalytic groups. And those catalytic groups actually catalyze the reaction. They cause bonds to be cleaved and bonds to be formed. And here are some features of the active site that are more or less common to all of the enzymes. So first active site is a three-dimensional cleft or crevice formed by amino acid side chains. So those amino acid chains or peptide chain in general in enzymes is globular. So there are obviously numerous alpha helices, there may be some beta strands, but overall shape is globular. And in that kind of rough sphere, there is a cleft or a crevice that is formed by amino acid side chains that point towards the inside of that crevice. And that's where actually the reaction occurs. The active site takes up a relatively small part of the total volume of the enzyme. And active site is a unique microenvironment. So it's hydrophobic. The chains that point towards the inside of the crevice or 3D cleft are hydrophobic. But there are some polar residues that are involved in reaction catalysis. Substrates bind to the active site by multiple weak intermolecular interactions. And of course, those interactions have to be weak. Otherwise, if they were strong, substrate upon completion of the reaction wouldn't leave the enzyme, and that would end up deactivating enzyme. And specificity of binding depends on the nature of the active site. So some enzymes are more, some are less specific. That depends on the nature of the active site. And when it comes to specificity of the binding of substrate to the active site of the enzyme, we have some models. Here we'll discuss a couple of them. The older model is lock and the key model, and that's an obsolete model. It's considered to be an obsolete model. It's analogy, or more of analogy rather than a model. Substrate is shown in blue and active site of enzyme in red. So in this case, there is a match between the shape of the substrate and that of the active site of the enzyme, and so they fit together like lock and the key and are held together through weak intermolecular interactions. As I said, this is considered to be an obsolete analogy or obsolete model. Much more modern and more accurate description is that of dynamic recognition or induced fit. And that means that substrate conformations of substrate and enzyme are not rigid. They have rather flexible conformations and they are in constant thermal motion. So they are not perfect fit for each other like lock and a key. Instead, they actually don't quite fit, but they basically explore shapes. They interact with each other and explore shapes and try to form intermolecular interactions. So they undergo thermal motion, form some interactions. If a few interactions form, then they may form some more, keep on changing shapes, and eventually that may result in a fit. Otherwise, if it doesn't result in formation of enough intermolecular interactions for them to be held together, then that means that substrate is not appropriate substrate for the enzyme and it's going to leave the active site. But otherwise, if it is appropriate substrate, eventually that will result that exploring of different conformations and formation of intermolecular interactions will result in induced fit, and they will form that temporary complex, enzyme-substrate complex. We have evidence that enzyme-substrate complex forms, and that evidence is direct and indirect. First, we should consider saturation effect. At constant concentration of the enzyme, the reaction rate increases with increasing concentration of the substrate until maximum reaction rate Vmax is reached. That means enzyme is saturated. Simply, it means that if we add small amount of substrate, then some of the enzyme reacts to catalyze the reaction, and reaction occurs at a certain rate. If we add more substrate, then more of the molecules of enzyme are involved in catalysis, and reaction rate is faster. And as we keep on increasing amount of substrate, more and more of the enzyme is used up to form enzyme-substrate complex, and reaction rate is faster and faster. But eventually, we reach saturation, Vmax, when all of the enzyme is effectively involved in reaction, and adding more substrate will not result in any additional increase in reaction rate because there is no more enzyme to catalyze that reaction. So all of the enzyme that is available is involved in formation of enzyme-substrate complex and catalyzes the reaction at maximum rate. So this is indirect evidence, something that we simply obtain on the basis of reasoning or rationalizing the observed dependence of reaction rate on the concentration of enzyme and substrate. We also have direct evidence. And direct evidence comes, for example, from X-ray crystallography, because X-ray crystallography has provided images of enzyme-substrate complex. So obviously, if we can see that enzyme and substrate form a complex, that's obviously a direct evidence. Also, forms of spectroscopy, such as nuclear magnetic resonance and electron spin resonance, provide spectra of enzyme-substrate complexes or transient species. And another spectroscopic technique is fluorescence spectroscopy. An example is given here. Enzyme tryptophan synthetase, it's also called tryptophan synthase, contains a colored prosthetic group, and that's pyridoxal phosphate or PLP. We will also encounter that one later in the course. So enzyme catalyzes synthesis of L-tryptophan from L-serine and indole triglycerol phosphate. And reduced activity of fluorescence is an indication of formation of enzyme-substrate complex. In this case, this would be enzyme-serine complex. And again, this is direct evidence. So this completes our brief introduction to enzymes.